prey, how they search for prey, how they decide what food items to, to eat, and how they make good decisions that lead to increased reproductive success, increased survival based on feeding responses. And it's kind of interesting. I was doing this at the beginning of my career, and now that I'm on the back part of my career, I started thinking about the other end of the animal and the decisions that animals make in terms of latrine habits, in terms of bathroom behaviors, and what when to excrete, what context, how much. And it turns out those decisions can have just as much impact in terms of survival and reproductive success as what's going on at the front of the animal. However, it's studied far less in general, I think, than feeding responses. Lots of predator-prey interactions, but less on that end. So that's what I want to talk about, and also talking about what information animals can glean from excretory products, metabolic products, including silk, to make better decisions about predator-prey interactions. So I want to also thank my co-authors here, Aslan Shulkusky, Amalia Esposito, and Zachary Klein, who were instrumental in putting some of this data together over the last 2 years. So, latrine behaviors matters, not just for spiders, but if you think about animals in general, it can be used to indicate ownership of territories. If you think of canids, if you forgive me for talking about vertebrates for a bit, I actually studied urine marking behavior in domestic dogs and discovered that it's a complex process. They don't just pee where they smell other dogs, but they make a decision 10 meters away. They make decisions based on the height of the urine mark, how many dogs went, whether or not it was a neutered or not neutered male, what other odors were associated with it, the sequence of dogs that peed there previously. So it's actually a complex decision process. So animals often make signpost signals, which is basically I'm here with their excretory products, and announce that. And that often has something to do with territorial interactions. And of course, regardless of the species of animal, feces, urine, other metabolic waste products are often transmission vehicles for pathogens and parasites. And so animals that are careful about where they excrete often have better outcomes. Another function is reducing the ability of prey to detect you. So if you're foraging, if you're looking for prey and they detect you before you detect them, it activates anti-predator responses, defensive responses that make it less efficient to locate prey. And of course, the inverse of that is also true. You don't want to alert predators to your presence with excretory products, smells that are associated with you, or indirect cues that can indicate your presence in the area. Probably some of the more famous examples of this in entomology, if you think of insects like silver-spotted skippers that projectile fling their frass, if I can say that, up to 30 body lengths away, and they found that they're removing these fecal pellets because if they are around the larvae, it dramatically increases predation from various wasp species, for example. So predator avoidance is a real key aspect of making good decisions about when to excrete and where. And in many cases, animals can actually get more information than simply the presence or absence of a predator, or presence or absence of prey, but actually detect what it is they're feeding on by excretory products. So, for example, if prey can tell that a predator hasn't just been in the area, but has actively been feeding on conspecifics, individuals of the same species, that tells them that it's a much larger predation risk than it otherwise would be, in which case they can grade their anti-predator responses proportional to the perceived risk. And so again, getting a lot of information from those cues. So animals make decisions about where to excrete. So for example, northern pike have bathrooms that they use. They have special latrine zones that they excrete in away from their hunting grounds. And the reason they do that is because they feed on various cyprinid fish that have alarm pheromones. So as those fish are being eaten, they're releasing alarm pheromones into the water, alerting other fish to the area that there's a predator and that they're likely to get consumed. And so when northern pike are feeding on these fish, every time they defecate, they're releasing plumes of alarm pheromones into the water that scatter potential prey for, in many cases, over an hour. And so it dramatically impacts their feeding responses. So making those key decisions about where to excrete really has impacts on feeding. And we think crab spiders do something similar to these northern pike. So Doug Morris did an interesting study with the crab spider Missamina vadia, and he would keep them in these little vials for up to 50 days, and he would feed them regularly, kept feeding them crickets or other prey items. And the spiders won't defecate for up to a 50-day period. And if you pull them out and set them on the edge of a leaf, 2/3 of them within 5 minutes will excrete over the edge of that leaf. So they're intentionally holding their excreta inside, presumably to avoid being or to fouling areas where they're hunting. And this is an area of research that we're working on right now in our lab. So crab spiders are amazing for their ability to hold their poop for long periods of time, well over a month. So spiders can convey information to predators and prey through silk and excreta. So I want to talk about silk too. So if you look out in the environment, there's the World Wide Web. I should say the original World Wide Web, or the internet of silk. This is a view out of my dining room window when atmospheric conditions allow you to physically see the silk in the environment. And it's everywhere. It's covering trunks of trees, it's covering branches, it's covering leaves, it's covering leaf litter on the ground. And most of what you see here in the middle are sheet webs. But there's orb webs, there's all kinds of silk. And this is a rich source of information for both predator and prey of spiders. So I want to give you some idea of how sophisticated the amount of information that's detectable through silk and excreta cues among and between spiders. So these are two spider species. On the left is Pardosa milvina, and on the right is Tigrosa helio. These two spiders are found very commonly in every corn and soybean field in the eastern half of the United States. Pardosa milvina can occur at densities as high as 250,000 individuals per acre, and they can eat 1 million insects per week. So not a small contributor to biocontrol within these systems. Tigrosa helio, about 50% of their diet is other spiders, especially wolf spiders. And so this is an extremely common predator of this smaller wolf spider, Pardosa. And you can imagine, because they're both wolf spiders, they're attuned to silk, excreta, and sensory detection systems are very similar because they're in the same family. So what we found is that not only does Pardosa respond to excreta and silk cues from the larger predator Tigrosa helio, not just identifying the presence or absence of a predator, but are able to discriminate between specific levels of predation risk based on information within that. So one of the things they do when they detect silk or excreta from these larger predators is they show this freeze response. They freeze up, they stop moving, and then they sort of tiptoe, making very slow motion through the area until they leave the area with that predator cue. They also show vertical climbing in the presence of these cues. But they're not just showing freeze responses or not, but they're showing different levels of those responses depending on what they're detecting. So we did a number of studies, and we found that Pardosa milvina, just using information from silk and excreta, can tell how big that predator is. Is it a juvenile? Is it an adult Tigrosa? They can tell whether it's a male or a female. Females are 4 times larger than males. They're more rapacious. They eat 4 to 6 times more food than males, so they're a more dangerous predator. And they show much higher freeze responses when they're detecting silk from a female predator than when they're detecting it from a male predator. They also can tell how old that silk or excreta is. They discriminate between silk that's fresh an hour old, a day old, or a week old. And they show increasing levels of freeze responses the fresher the cue is. They can tell the hunger state of that predator. So they discriminate between silk and excreta from a spider that hasn't eaten in 2 weeks compared to one that's just eaten. And they show the strongest response in the presence of cues from a hungry spider. They can tell predator species and respond based on risk that way. And just like the northern pike and cyprinid fish, they can tell the diet of the predator, whether or not they've been feeding on Pardosa or if they've been feeding on other prey types. So lots of information that they're gleaning, and they're calibrating their defensive behavior proportional to the perceived risk. So pretty sophisticated system, and this is why it matters what the latrine habits are of predators. So just to give you an idea of how we measure this stuff, we'll give you an example experiment. So testing whether or not Pardosa can detect the diet of Tigrosa based on excretory cues and reduce activity in an adaptive manner. So in this case, we might take filter paper and allow a Tigrosa, the larger predatory spider, to deposit silk and excreta on that cue. And we might do this with Tigrosa that have been fed nothing but crickets for 2 weeks. And then we might have another set of Pardosa, or Tigrosa rather, that we fed nothing but a diet of Pardosa for 2 weeks. So the only difference is the diet cues that they've been feeding on. And then we drop a Pardosa in the center, and we let them walk around on these different substrates, and we measure their activity levels based on what kind of silk and excreta is present. And when you do this, you get a big difference in freeze responses. So Pardosa shows much less activity, more immobility, more careful tiptoeing through the paper when they're detecting Tigrosa that have fed on Pardosa compared to ones that haven't. So this is the kind of data we collect. Now, after you demonstrate that you see a difference in behavior, that they show greater levels of immobility or freeze response, we wanted to test whether or not that actually translates into increased survival in the presence of a live predator. So we did the same experiment, but we then added a live predator that had been controlled for hunger level. And when you do that, you can see that there's differences in survival of Pardosa based on what kind of chemical information they have available to them. So if you throw them on a sheet of paper with a predator but no other cues, they all get eaten really quickly. Most of them are dead within a few minutes. If you place them in a container with cues where Tigrosa-fed crickets have been, they survive longer, and they survive even longer if you put them on a substrate with cues from a Tigrosa-fed Pardosa. So those graded periods of immobility translate directly into differences in survival in the presence of a live predator. So it's pretty effective anti-predator response. So that's an example of the kind of studies we do to parse what information they're gleaning from spider predator, excreta, and silk. One more experiment, just to give you an idea of how sophisticated the system is. We discovered that Pardosa can assess predation risk and detect what I would call as kill zones left behind by Tigrosa helio with silk and excreta. In other words, can they tell when they're walking to an area where Tigrosa has recently killed another Pardosa compared to interactions with other prey types? So thinking about it this way, we know that they show an anti-predator response, a freeze response, when they detect silk and excreta from this larger predator. So the question is, if Tigrosa helio is actively killing Pardosa, does that translate into different excretory products, different silk deposition patterns, maybe a stray leg from an eaten Pardosa, and does that translate into an elevated anti-predator response? So are the spiders able to discriminate between these kill zone areas versus ones where they're simply detecting a predator in the area. So the way you can test that is you set up an arena, a control arena with just a blank sheet of paper. Then you can have another set of paper where you allow conspecific pardosa to lay down silk, which should be a low level of threat. You can have another environment where you have tigrosa and pardosa depositing silk, but there's no prey consumption. So you basically move pardosa around under vials and move the predator around under vials. So they're both depositing silk, but there's no predation. You can throw tigrosa in a container with crickets and let them eat them. So there is predation, but not of pardosa. You can have a treatment with just the predator and their silk and excreta, no predation. And then critically, tigrosa with pardosa added and allow predation, and then take the spiders out of these different arenas and just put pardosa in to see how they respond to these different substrates. And so when you do that, you get this gradient of immobility. So they show the highest mobility on a blank sheet of paper, and they show the highest level of immobility or freeze response on the substrate with tigrosa and pardosa cues where tigrosa has been feeding on pardosa. And if you do the same experiment here, where you introduce a live predator on those different substrates, live tigrosa on those different substrates, and you add a pardosa, and you look at how long it takes before pardosa gets eaten, they survive the longest on these substrates where they're detecting silk and excreta in an area where tigrosa has been actively killing pardosa compared to areas where they're not, where they show much less freeze responses and much less predation. So again, I just want to point out there's a lot of sophisticated information about size, sex, species, diet, hunger level that prey can extract from predators from essentially decisions they're making about metabolic waste products that they're leaving behind. Okay, so I've been talking to you about all this just to convince you that prey species have evolved really sophisticated systems for detecting excretory products and calibrating different levels of perceived risk and allocating defensive behaviors in an adaptive way to avoid being eaten. So then the question is there a way that predators have evolved a counter adaptation or have co-evolved counter responses to overcome these prey responses to their cues by either changing their latrine behaviors or in some way manipulating prey behavior with that information? And so we ask this question, are these excreta-mediated defensive behaviors of prey, can they deploy them in some adaptive way? So we're going to move away from wolf spiders and move to a very different spider species, the purseweb spider, Atypus karschi. So purseweb spiders are interesting in a lot of ways. From a phylogenetic perspective, they're sort of close to the ancestral spider. They're atypical tarantulas. They occur in parts of Pennsylvania. There are several different species. So we do have tarantulas in Pennsylvania. There are also tarantulas in Maryland, although I don't think these are in Maryland. But they spend their whole life inside of a tube that's roughly the diameter of the spider itself. So these are vertical tubes that they attach to vertical substrates, and the tubes are 16 centimeters to about 20 centimeters in length. It's almost like a floppy sock that they live in. And half of that tube is above ground level, and half of that tube is below ground level. And then they just move up and down that tube for their whole life. These spiders live up to 10 years. I've collected them in the wild as adults, and I had my last one die just a couple weeks ago that I collected in 2018. So they definitely can live for many years, and they don't leave the web except to add bits of material as camouflage on the outside, or it appears they seldom leave. So the way they get food is they're extremely passive feeders. They sit in that tube waiting for an insect to walk along, a walking insect to walk along and hit this tube. And when they start moving on this tube, the vibrations set off the spider. It bites through the web with these extra long fangs, grabs the prey, and drags it inside lightning fast, and then seals off the web. It's so fast, it's almost difficult to see the prey. It's there 1 second and it's gone, and you never see the spider. So they're always inside. So a spider that never leaves this web, hangs out in it for 10 years, begs a few questions. So one is, this is a low surface area web. So it's not like a big orb web where there's lots of surface area. There's no glue involved. They're essentially waiting for some insect dumb enough to bump into their web and they get food, which to me seems unlikely for them to get much food that way. So we had a few initial questions. One is, how do these spiders capture enough prey with this very, very simple, very, very small web that they never leave? And if they're living in it for up to 10 years, it creates an immediate issue with hygiene problems. So we're talking about latrine habits. They're excreting for 10 years from this single tube. And how do they do so in a manner that they're not littering the area of their web with so much excreta that it's repelling prey that might otherwise walk into it? So these were some of our initial thoughts about this. And we know that prey species are paying attention to excretory cues, certainly. So if you think a little bit about where these spiders live, they're often up against walls or tree trunks or other vertical structures. And it's not uncommon to find them nestled sort of like between root masses, something like this. And in this situation, it isn't hard to imagine where they're using those environmental walls as a way to kind of funnel prey to the web. So you can imagine a cricket or some other ground arthropod would hit this tree and show a thigmotaxic response, which is wall hugging. Most animals will hug a wall and avoid open areas, and then move towards the web and be caught. However, so if this is the case, they might be using something like a tree or a wall as something like a drift fence. So a drift fence, for ecologists, is a way to trap prey and take advantage of thigmotactic responses. So the fact that animals, when they hit a wall, they end up walking along it until they find a way through. And in this case, you set your trap there and you would catch, whatever your small mammal is. And of course, entomologists and arachnologists will make various drift fences with a pitfall trap in the center to catch ground beetles or whatever. And it's the same idea. So we see these kinds of drift fences, and we would say, well, perhaps these spiders are choosing locations where the natural structures of the environments around them act as a drift fence. However, it doesn't solve the issue of excreta, so the hygiene problem. So are they randomly defecating in the area? And if that's the case, it may repel ground arthropods and cause issues. So we hypothesized that maybe these spiders are excreting in a non-random way. They have a precise directional location for their excretion, and that they do it in such a way that it doesn't create a large repellent zone of excretory products of a predator. So in this case, maybe they're depositing excreta sort of over the top edge here where it wouldn't be in the way. Or they're depositing excreta underground and removing it in some way in that case. But either way, we suspect that they're careful about their hygiene. Now, this story works pretty well when you talk about these spiders along vertical walls, fence posts, tree trunks, whatever, root masses, exposed roots. But we also find these spiders out in the open. So in an open field, and they'll attach their purse web to a blade of grass. And so when they're in that environment, there aren't natural features that readily lend themselves to a drift fence or a way to funnel prey. And so you can imagine, again, if they're randomly defecating, that they're creating a repellent zone around that web. So we hypothesize that if they are non-random in their excretions and are able to direct them in a way, they could potentially make a drift fence with their own excretions if arthropods, when they encounter this, treat it like a drift fence. If they hit it and walk along it instead of willing to cross over it. So this is again, interesting idea that we're interested in testing. So we also thought they could use sort of these excretory drift fences, even in cases where they have vertical structures. So they could lay down a linear array. And so if prey hit this, either the roots in this case, or hit this excretory line, they're still going to have a 50-50 chance of turning and moving towards that web instead of away from it. So this is kind of what we're interested in testing. Our specific questions: what are the latrine habits of this odd little spider that spends its whole life inside of this tube? How frequently do they excrete, and where? Is it outside the web? Is it inside the web? If it is outside the web, what's kind of the spatial pattern of excretion? Is it like a single point? They launch it like a catapult and it drops at one location. Is it linear? Is it a multi-vector? Is there a pattern? What might it be? And is it non-random in its direction? We wanted to test that as well. So another question is, since they're out in the open fields and they're also up against vertical surfaces, are their excretion patterns different when they're against a vertical wall, horizontal wall, compared to if they're out in an open arena? So that's one set of questions about the latrine habits of the predator. And then the other question is, what are the prey doing? Do purse web prey modify their behavior when encountering purse web excreta? And is it possible that these excretion patterns function as a drift fence trap for prey? So to answer these questions, we went out to Tyler Arboretum in Media, Pennsylvania, down in southeast Pennsylvania, and we employed the services of Stephen Tesler. Steve Tesler is fantastic. He is the purse web guru, knows more about these species than just about anyone, and is really good at a search image to identify where they are and how to collect them and how to keep them alive. So he's been a tremendous help in this regard. A little bit more about this species. It was originally described as Atypus snetsingeri, and it was described as the lone representative of this genus in the United States, in fact, in all of North America. And so it was this bit of a taxonomic oddity. And it turns out, in fact, Steve was one of the co-authors that discovered that it is, in fact, the same spider species as ones from Southeast Asia, Atypus karschi, and that at some point in the past, they were imported into the United States. We suspect in botanical specimens, particularly they were close to this arboretum and were introduced in with the soil materials, and nobody noticed. And then they spread out from there into parts of Pennsylvania. But they're very slow at spreading. They are not spotted lanternflies. They are pretty localized in their distribution. And here's a couple examples of these purse webs just in the vegetation. So we went out there to collect these spiders, and it's much like digging up a plant. that you want to bring in. So you take a shovel, you dig a big wide area around it, and then you slowly start to excavate until you get close to that purseweb. And so you have to chisel away with a spoon. It's almost like an archaeological dig until you have this sock of a purseweb that you then can transplant with the spider in it, and then it will build a new web. So two of my co-authors here, Zach Klein and Amalia Esposito, were helping in this process, digging up these spiders. So it felt like gardening more than spider collecting. Once you bring them in from the field, you put them in these 32-ounce deli containers with some dirt in the bottom, and they very quickly build these pursewebs. Here's a brand new clean one before they put various forms of camouflage on it. And they answer the question about where they excrete pretty quickly. Within a day, you could see excretory products showing up on the walls of the container. So we wanted to see where they were defecating in the containers relative to the location of the purseweb. And this shows you how elaborate some of the camouflage can be on these. They put all kinds of bits of soil or whatever that they build up around it. Other ones are more Spartan. And we wanted to see if it was non-random. Here's a little bit of excretion here from spiders. But we also saw silk around it, suggesting that they do mill around maybe in the middle of the night. You almost never see them. You never see them out during the day. And really, I've never seen them out, period, outside of their webs. You have to check and see if they're alive periodically, except for crickets disappearing, you don't know. So we measured the excretion. We took all these pursewebs and we turned them so that they were facing south, and the attachment point was due south. So we reoriented all the pursewebs, and then we measured excretion patterns. And when we did this, we found that they all sort of defecated, at least this particular individual defecated in a northern orientation. So this is a rose plot. It's like a round histogram. So if you imagine looking down at the container that the spiders are in, the length of this arm is how many excretory events occurred at that angle. So you can see the majority of the angles were here in the 330 degree to 345 degree range. And this black line's the mean. And then this is the standard error around that, just to give you some sense. So this is 19 defecation events for spider AK44, and it showed significant directional orientation. It wasn't random. Another spider, broader range, but also non-random, and more or less the same direction, north by northeast, or northwest, sorry. For this individual, 17 excretory events. Here's one that is much more precise in where they're excreting. 11 excretory events. So you see this pattern forming across these different species where they're all excreting directionally, non-randomly, and more or less in the same direction. So if you combine all the results, so this is 680 excretory events from 46 different spiders over multiple months, and you get something like this where the mean heading is almost due north. So all these spiders are essentially doing the same thing, and they're using the same rule for excretion. So it's non-random, it's a single vector opposite the attachment point of the web. Highly significant, non-random. You do see some events, around this, but the vast majority of them are one direction. So 43 out of 46 spiders were non-random in their orientation, their excretion orientation, and 41 out of 46 were excreting north, significantly, due north. So that was interesting, and that's what's happening inside of a container. So just visually, it looks like this. This is 6 days of housing in a container. Here's the spider web. There's a few excretory hits along the wall here, but most of them are due north. So that's what that looks like. Then we asked the question, what are they doing out in an open field? Now, this is a spider. I had to pull it out of the web to take its photo. It does not like to be in this environment at all. I should say this is a pretty artificial, I mean, it's a real spider photo, but not in their natural condition. If you want to try and photograph them with an endoscope, you put an endoscope into the purseweb and they take their chelicerae and bite it closed and prevent you from actually going down into the tube. So they're maddeningly difficult to film and see what they're doing inside the tube. So you pull them out, you have to get their picture. So we asked the question here, what are they doing in an open field? That's a harder question to answer. So we had to be creative. And what we did was we cut down the sides of the container so that the spider didn't have to build a new web. And they got to keep their web, and we could change the orientation of it. And we put it inside of another deli dish as a sleeve so we could pull it out or not. And then we took this cutaway and put it inside of a kiddie pool, a pretty large kiddie pool, 1.5 meters, a little over that. And we have little cotton balls in the corners that were moist in case the spider happened to get out in the middle of the night or something, and we didn't want it to desiccate. So we set them up in essentially the same way, and then we measured excreta this way in terms of directional orientation. Now remember, if they're using a drift fence, we're expecting excretory patterns to look either like this on the left or like kind of a cross pattern if they're using a four-way drift fence, two-way or a four-way drift fence. So that's what we were sort of looking for to see if that's what they do. And we had several rooms set up like this with kiddie pools everywhere. We bought out all the kiddie pools in central Pennsylvania, and we had to keep buying bigger sizes because we couldn't record all the excreta that they were depositing and catching it all. It would go past the range of the pools. So this is Aslan Shilkuski, who's taking measurements of spider excretion in these pools, in open arenas, in the center of open arenas. And when we measure this is 46 spiders with 82 excretions. We get a very clear answer, and that is these spiders produce a bidirectional, sort of 180-degree angle, two vector excretion lines that are opposite of each other. And they build those up over time. So we see this highly significant pattern, non-random distribution. And notice it's different. You take the same spider that poops in one direction in a container, and now it's doing it in this axial direction when you take that same spider and put it in an open arena. Now, some statistics here. The mean length of their excretions was about 0.5 meter, 54 centimeters. The maximum minimum distance before the excreta hits and extends out past that was 64 centimeters. So quite a distance from the tube. And we got one spider that excreted a distance of at least 1.2 meters from their web. And when they're excreting, they're sticking their abdomen up through the top of the purse web and shooting directly from their anus. So they're not flinging it. They're not using their legs. It's presumably hemolymph pressure that they're using to generate that. So my students call these ICBMs, intermittently catapulted bowel movements. So, they're catapulting these lines, these linear lines. So when we did the math on this, it appears, and I would love to hear a counterpoint to this, that this is the longest projectile defecation for an animal relative to body size. So there's your dinner conversation for tomorrow. So these are 2 centimeter long spiders. And
did you call Guinness, Matthew?
Well, maybe I should. So they're defecating 60 body lengths. So that's equivalent to if this was a human, you're standing in the end zone and you're defecating and it's hitting the other end zone. So it's an impressive length.
That is impressive.
Yeah. So even longer than the silver skippers, which are about
Hmm.
30 to 35 body lengths, I think. So I'm sure a functional morphologist would love to find out how exactly they do that. But you have to wait 3 days to film it. And I've tried, it's hard, but anyway. So this is interesting. And I'm pretty sure that they can actually go further than that. It's just their excreta is hitting the edges of these kiddie pools, and we couldn't find big enough kiddie pools to actually record it and do multiple replicates in lab spaces. We had to use actual pools, I think, to document the actual distance. So so the patterns look very much like or consistent with a drift fence. We then wanted to see how prey respond, and luckily, it's fairly easy to find out what purseweb spiders are eating because they leave their carcasses, the remains of their prey, at the bottom of their pursewebs. So as you dig them up, you'll find like millipedes, sometimes centipedes, but a lot of millipedes, and a lot of woodlice. So woodlice seem to be a common prey item of these spiders, and for that reason, we were interested in testing how woodlice respond to purseweb excreta. So we grabbed some Porcellia laevis, woodlice, pillbugs, roly-polies, all the other common names, and set them up in these kiddie pools. So we allowed spiders to poop up the place for 16 days, and then we put in different sectors. So we have an outer edge sector, which is kind of this trough around the edge, and then we have an outer sector, middle sectors, inner sectors, and then the place where the spider web is. And here's one of the woodlice for scale. And also you can see just 2 excretory lines here. This is just 2, not 16 days worth. And you can get a sense of the distance. They start to go up the sides of the container, but they're these lines, this kind of spackle lines that are set up away from the web. And so we were simply measuring, where do these pursewebs spend their time? So we tested them under 2 conditions. We had a control kiddie pool where there was no excretion, there was no spiders previously in it. And then we had the ones where a purseweb spider was hanging out in the center for 16 days, depositing excreta. And then every 3 days after they pooped, we take them back, put them in a sleeve, feed them, and then put them back in so that they had, never-ending supply of excretions to put in the container. So when we test that, we get results that look like this. So this is the number of sectors entered by the woodlice at least once. So if you look along the edge, not surprisingly, they show a strong thigmotaxic response where woodlice like to walk around the edge. But in the containers with purseweb excreta, you see a pretty dramatic and significant drop in how much time they're spending along the edge. And they're spending significantly more time in the middle sectors and even the inner sector where that purseweb is. So they're actually moving towards where that purseweb is when there's purseweb excreta present. And you can see the same pattern, the total number of sector visits across sector types, same pattern. They're spending more time in the center. They're visiting those center sectors more frequently than they are the edge sector. So at least with woodlice, it appears that pursewebs are using excreta in a way that is funneling prey towards their webs in an adaptive way. So we didn't find any evidence that they were excreting within the web. We would take apart the bottom of the web and there wasn't anything there. So they're definitely excreting only outside of the tube, and they're doing it between every 1 and 3 days, depending on how much they're eating. And importantly, we saw different strategies for excretion for latrine behaviors when they're in a closed container, or if you put them up against a long wall, they will excrete perpendicular to that wall. And then if you place them out in an open arena, they will excrete bidirectionally relative to their position. And then they'll go back to that. So this appears to be the case on an individual basis. So individual pursewebs are using this system, but it looks like the entire population of purseweb spiders are following the same rule for excretion. I mean, if you turn them so that their attachment point is a certain direction, you could reliably see them defecate in a different way. So in open arenas, you have this bidirectional area, and that also is fairly consistent across pursewebs. And impressive in terms of the distance. So again, projectile defecation more than any animal. I remember I saw an article about penguins, and they amazingly could defecate 4 feet away from their body. And I was thinking, well, that's nothing compared to these guys. So pretty impressive. The average is 30 body lengths with the maximum of 60, but it wasn't uncommon for them to be 40, 50, 55 body lengths distance. And then, of course, the wood lice were more likely to move towards that purseweb when spider excreta is present than when it's not. So we're saying tentatively, and again, this is only one prey species. You could test this with spiders and with crickets and with millipedes and other species to see how they react. And it may be that different prey react differently, which may impact what type of prey they're actually feeding on, ones that are susceptible to walking along an excreta line versus ones that aren't. But their behaviors seem to be consistent with using excreta in an adaptive way as a drift fence to take advantage of prey's avoidance of predator excretory products, which I think is really interesting. And if this is the case, then it looks like the ecologist's invention of the drift fence probably was an invention, and that these spiders probably invented it as early as 200 million years before any of us thought of it, which also is pretty impressive. So I'll pause there. I just want to again acknowledge Stephen Tesler for his help on all of this, Susquehanna Research Partners program that helped pay for student stipends in the summer, who helped work with this project and during the academic year. And I'll stop there and ask if there's any questions.
I was curious about the purse that they make, and this is all made with spider silk, is it?
Yes. I
Is there
don't know. Oh, go ahead.
any material stuck to it at all to disguise it?
Yes. It varies in the amount of disguise that they have. Some of them are extremely cryptic. They blend in, they use twigs, some of them use sand, pebbles, uh, bits of leaves that are cut up and posted like mosaics. So it's really variable. And then there's some that don't seem to care about camouflage, and you can see the silk. It could be that they're more recent or that they've added silk to it, but really variable in the types of materials they attach to it. Non-sticky. Now the inside of the web over time gets almost like leather. It gets very thick and it's very smooth, which makes me think it's multiple silk types. So they have multiple silk glands that they may be using combined. So it may be a composite silk. I don't know. But the first couple days when they make it, it's this kind of standard webbing that you would see with a funnel web or some other spider, but then it thickens and they keep adding to it and adding to it until it's this very smooth material that you have to take scissors to cut. It's tough to open.
Hmm. Wow.
Yeah.
How big is the spider?
Adults are about 2 centimeters long.
Okay, so they're not real big at all, but
No, they're not too big, but for an animal that poops them 1.25 meters, it's pretty good. Um,
yeah.
uh, and they can capture prey that's pretty big. And
hmm
if you see their fangs are extremely, chelicerae are extremely long, and that's to puncture through the web and pull it through.
Yeah,
I
I
didn't
see.
talk. Oh, go ahead.
I was thinking they're reminiscent of the Sydney funnel web spider. Is there any relation at all? I mean, it's too far away, but I just wondered if that was that their basic anatomy similar in that regard.
Yeah, these are pretty basal on the spider evolutionary tree,
Okay.
atypical tarantulas. Um, uh, and they have some interesting habits. I've collected other species, Sphodros niger. In fact, my original thoughts about this project in some ways date back to graduate school. I had collected Sphodros species of purseweb spider and put it in an aquarium on my desk in graduate school. And there was spider excreta showing up on the other side of my desk. And I eventually figured out where it was coming from. I put a lid on the aquarium, and that's where it was coming from. And I was like, I cannot believe that spider poops that far. And I kept that in my head for decades, um, and asked the question with this other one. So I suspect that I'll go on a limb here and say that all purseweb spiders are capable of ballistic projectile defecation. Whether or not they use the same system or not, I'm not sure, but they certainly are capable of launching their ICBMs. What one other aspect that's curious about this spider, a little natural history. Um, so males, once they reach sexual maturity, they tear down their purse web. They destroy it completely and down to like where the hole is, and then they wander off. And if they're in a container in my lab, they spin around in circles endlessly until I take them out and put them in with a female purse web. And when he climbs out, we managed to see a couple. We actually couldn't see the matings, but we saw the courtship where males will climb up the side of the purse web of a female and they'll drum on it with their legs. They'll drum and then they'll back away and drum and back away. And every once in a while, you'll see two fangs come through from the female. And he'll drum more, two fangs. You don't see the female. And then at some point, the male, maybe it's a vibratory signal from the female, he rips open the purse web and darts inside and it seals up. They seal it up. And that's where mating happens. Now, I assumed based on other spiders that male mates and then he becomes a meal. He gets eaten by the female. But I excavated one of the webs about 6 months later, and that male was still inside, cohabiting with the female. And there were spiderlings inside of the purse web. So I don't know if that was atypical or not, but it appears that um, you know at least cannibalism doesn't always happen, and maybe it never happens. I don't know. So I thought that was an interesting bit that the males just give up their web and move in with the females um, for mating. But that's really the only time they leave the web for long periods of time, other than to decorate the outside of the web. Or at least that was my experience watching them.
Matt, I had checked the Maryland Biodiversity Project to see what species of pursewebs we had down here, and they show three species of Sphodros. Would the the webs be similar looking to the ones that you've have found?
Yes, they look similar. Um, yeah, purse webs require training your eye. They are hard to see, but they can be super dense in certain areas, and they're locally distributed. So you'll find nothing, 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 and then here's a bunch of them, which makes me think they just don't disperse that far. Um, um, I was at Fort Indian Town Gap. Mark Schwartz is there, and I was collecting, doing survey work there, and we caught a Sphodros at that location. Um, there's also trapdoor spiders, Antrodiatus, which I happened to find one in my garage of all places. It's the only time I found one. Um, and so apparently they're living in my yard. And as many times as I've looked for spiders, I've never seen these trapdoor spiders. So there's other atypical tarantulas around, and that's moving towards the northern part of their known range. Um, so I think Maryland, I'm sure has those spiders as well, and they're just maddeningly difficult to see. They have, you know, trap doors that perfectly blend with the background. There's a few people I know that are able to spot them. I'm not one of them. Um, but they're uh, they're interesting spiders for sure.
What kind of habitat are we talking about to find one of these things?
Um, the Atypus species are real generalists. You know, Sphodros, when I found Sphodros, I've typically found them in deciduous forest, and find them on trees, around the bases of trees. Although I haven't looked beyond that, I think it's just easier to see them there. Whereas the Atypus species here, they seem to be all kinds of places, open fields, even mowed fields that have been cut periodically a couple times, 3 times a year, 4 times a year. Um, and they're also found in deciduous forest. They're found in edge habitat. They're found mixed in with poison ivy, unfortunately. Um, so they don't seem to be that fussy about habitat. You can find them down around riparian zones, close to rivers. Um, so
I guess what I also wondered, I mean, are we talking about Piedmont or are they found in the mountains or I mean, can you even see them on the coastal plain?
uh I think so, yes. Yes,
Okay.
to both of those. One curious aspect that I didn't get into much is, so I've been talking about these as vertically oriented webs, but there are some that build these horizontal ones across the ground. And I've seen some, I don't know what percentage of the population does that, but that brings up other questions about, how they're excreting. But I think there's alternative strategies for prey capture in some of these spiders. And the other question is, what are they doing underground? Are they feeding on, are they catching earthworms that bump into it? I don't know. So uh, there might be a whole nother, uh, you know, food type available for underground prey or above ground. I don't know. So they're a difficult species to find. They're not uncommon, but they're difficult to find. And just as a behavioral ecologist, they don't want you to watch their behaviors. They shut themselves off, and so excretion is one of the few things you can measure easily without destroying their web.
Anybody else have some questions they want to ask? Shoot now or forever hold your peace. Well, all right. Uh, Thank you for answering my questions. I appreciate that. And thank you very much for the presentation. That very unique kind of presentation, I have to say. Uh, it isn't something I knew anything about, honestly, so it's very cool hearing something completely new to me. So that's pretty neat. Good stuff.
Well, they're new to
Um,
me too. I'm used to wolf spiders, and this is a side project. So, um, yeah, no, not many people. There's James Bond, not James Bond, Jason Bond, who studies Atypus and did the taxonomy on them. But beyond him, there aren't a whole lot of other people working with that group.
you're very specialized. Yeah,
Yeah,
indeed.
absolutely.
Well, I mean, it comes down to that with a lot when we studying arthropods. I mean, there's specialists that are, three in the whole world or something for a certain taxa. That very seem to be often the case, with entomology anyway. So, um, anyway, but thank you for your time joining us tonight and
Sure.
and bringing the presentation. It's pretty thorough study you did there, figuring all this out.
I have one question. Can you find these webs at all times of the year or just certain seasons?
Um, that's a good question. I haven't looked. I mean, that's a Steve Tesler question. Um, I think they're up year round. Um, I think you can find them year round. Now, Steve talked about ballooning activities, which is interesting because in
Hmm.
the spring, there's this short period of time in the grassy areas where they are, where the spiderlings will climb. They'll pick. I don't know if they use cues from each other, like silk cues from each other, but they pick a stalk. It could be like a goldenrod plant or something, and they climb up it, and then they try and balloon off, but they don't seem to be that great at ballooning, and so they kind of make a tent over these goldenrod plants. So they make these kind of teepee shapes um, over them, and then they land, and that's where they set up shop. So you can see these fields full of dispersing Atypus spiderlings just by looking at the plants. But it's this narrow window in time that it happens, and that's where you can identify dense populations of them. So again, probably not good at dispersing relative to other spider species.
I was curious since we have a deciduous forest directly behind our house. I was wondering how easy it would be to, it doesn't sound too easy to find them at all, but
If you see one and know what it looks like, you can find them, and you're like, oh, there's actually quite a few around here. But it's finding that first one that's
right.
that's that's hard. And again, they're not terribly uncommon, but just hard to locate. Having the search image is hard.
I'll be on the lookout. I live in an area where I have a riparian ecosystem right in back of my house, and um, big trees, small trees, all kinds of, those slopes. So I'll keep an eye out. I guess they're throughout the year, I would imagine, but they must be sort of dormant in the winter, I would guess, right?
Yeah, they're there. They go underground, and I don't even know. I mean, there's so many spiders. We don't know what they're doing in the winter. Um, I
Hmm.
suspect a lot of them super cool. Build up sugars in their hemolymph so they don't freeze and hunker down. But not really sure.
Right. Okay. Well, um, I guess we have your contact information, so we can be in touch with you, and we have some things to send you again. Um, you'll get a journal from Jean and that sort of thing. Um, thank you again, and good luck with the teaching over there at Susquehanna. Are you going to hang on for a while teaching?
I think so. I don't know. We'll see. I'm hoping to put in a little more time. I'm working with 11 students now on different projects, so I've been keeping busy for sure. Um, who knows? Maybe I'll talk about fishing spiders and um, or wolf spiders and what effects they have on questing behavior and ticks and things like that. So we got all kinds of stuff in the mix right now.
What did you say about the ticks again?
Um, what I have a student looking at larval ticks, questing behavior, and how they respond in the presence of wolf spiders. So, we've been talking about silk cues and um, excreta cues. And so when ticks quest, they come up to the surface and climb things to latch on. And that also makes them more susceptible to predators. So we're kind of looking into that. Spider tick interactions, if there are any, who knows? So those are some some of the stuff we're working on. We just published a paper today in the Journal of Insect Behavior um, on Dolomedes triton, which is the most aquatic spider, one of the most aquatic ones, and how they put hydrophobic silk across the surface of water, um, ponds, rivers, and um, Neustonic prey, water striders, some wolf spider species, detect that and they change their behavior. Water striders get out of the water when they detect this silk from these fishing spiders. So we're investigating the behavioral interactions of that as well, whether or not they're using it as like an enemy avoidance pheromone. Um, because it looks like the silk's used in male female interactions, but water striders are kind of eavesdropping on this sexual communication in spiders, and they're changing their behavior. But the silk actually increases um, predation success of Dolomedes. So we think that the silk might increase the sensory detection range of the spiders across the water surface like a trip line, and it alerts them to where all these prey are on the surface. So that's a paper that just came out in Insect Behavior today, literally today.
Wow, pretty
It's
cool.
kind of fun. If you want to check that out.
Well, maybe we can call on you down the road if you'd be willing to talk again sometime in the future for us.
Sure, absolutely. Yeah, I've been doing research on um, pesticides and spiders, lethal, sublethal effects, things like that.
Hmm.
And we've unfortunately have a lot of pesticides that have really negative consequences on spider populations and agricultural systems, um, like bees.
Like everything.
Yeah, like everything. So it's pretty devastating.
Yeah,
Yeah, that's a sadder story to tell, but
but it's a true story,
yeah,
unfortunately. Yeah.
yeah, an important one, I think. But yeah, this is great. This is fun. I love these society driven journals too. I think um, it should be the model for all scholarship in academia, instead of the large corporations that run academic publishing and things like that. Um, so
That's nice of you to say so because I think there's a lot to be said for that. Absolutely.
Oh yeah. Yep. Yeah. We do the Journal of Arachnology, which is sort of the spider, spider equivalent.
Right. Yeah.
Yeah. But again, it's society driven publication.
Yeah,
Jeff Schultz, I think, is a member, isn't he, of Maryland Entomological?
Ted,
Jeff Schultz.
Jeff Schultz, yes. Yeah, he's a member.
So he's a great functional morphologist. He of anyone, it seems like, would be primed to figure out how they're able to shoot excreta that far.
Mhm.
Find somebody who's able to do it. My physics isn't good enough, I don't think, but
Pretty neat.
yeah.
All right. Well, we don't want to
Okay.
keep you
Very
too
good.
long anymore. I mean, we may talk a little business for our meeting, but certainly, if you want to stay on, you're welcome. If you want to get out and go grab some dinner or something,
Yeah, I
that's
think maybe,
okay.
but it was great. It was fun.
Yeah, thank you, Matt. I appreciate you talking tonight.
Yeah,
very
thank
good.
you. Thank you all.
Thanks again.
All right, thank you.